Hello and welcome to Corning Life Sciences presentation. My name is Laurent Picard and today I will be introducing a new adherent cell growth technology for bioproduction. I think most of you are familiar with Corning Life Sciences lab consumable products and our highly recognized brands with a reputation for quality, such as Falcon, Axygen, and Pyrex. During 100+ years of history, we've built a strong core capabilities in cell culture, and those capabilities are now poised to help Corning and the market address the challenges presented by bioproduction and specifically by cell and gene therapy manufacturing. One of the methods of choice for viral vector production is adherent cell culture scale-up in multi-layer culture systems, such as CellStack, as shown here, or more recently in other Corning innovation, the HyperStack. These systems are easy to use and able to produce billions of adherent cells, but scaling beyond 10 to the 16th viral genome range has been difficult to achieve, as 2D systems take a lot of GMP room and suite space, are quite labor intensive, making them non-practical for very large viral vector production for large indications. Our mission has been to change this paradigm and enable bioproduction facilities to build faster and more cost-effective scale-ups of adherent cell culture. As the demand for viral vectors continues to rise, we now need to provide customers with a solution to the level that they need beyond the HyperStack and the CellStack scale-up challenges. The technology behind the Corning Ascent Fixed Bed Reactor, or FBR, is our solution to this, and it has been specifically designed for bioproduction. We have optimized performance for high cell density growth, leading to improved yields, driving significant cost reduction with true scalability from process development to production from 1 square meter to 1,000 square meter. We have validated the concept of 3 times better viral genome yield compared to existing FBRs, not only in our labs, but also with some of our early partners. And we are launching this technology in the first half of this year, starting with a process development unit, as shown here, that can be operated in a closed system fashion with connectors. It will be for process development, simple to operate, compact, and easy to use. More importantly, it will help us continue to validate the value and move to larger sizes and production scale bioreactors. The Ascent FBR provides significant economies of scale compared to existing technologies such as HyperStack and CellStack. This is what this table shows here. As you can see, the 5 square meter process development scale FBR is about the size of the 20-ounce can and is the equivalent in surface area to about 3 36 layers HyperStack or 2 CellStack 40. This demonstrates the significant surface area intensification for large-scale adherent cell culture. The material design of the Ascent FBR is what drives the yield improvements. As we will see in subsequent slides, the 2 key features of the system are the uniform fluid flow in the bed and its consequence, a very homogeneous cell distribution leading to uniform growth. We've also designed the system so that it would be truly scalable and would perform the same way from small process development size to larger 500 or even 1,000 square meter. In order to do that, you need to be able to harvest and transfer cells to another vessel in a sterile or aseptic way. We'll show data on this as well and how this enables better yields compared to competition, lower media consumption, and reduce time to scale up. So how we've done it. Concept is the packing surface area in a small volume. That is not something difficult and has been done before for other FBRs on the market. What is hard, though, is to have the surface area uniformly distributed throughout the vessel, and consequently having homogeneous fluid flow and distribution of cells throughout the mesh bed. The substrate that is shown here is a woven PET mesh treated to enable cell attachment and packed in the bioreactor vessel. Cells behave uniformly, stay healthy, perform better, are transfected better, and eventually this leads to higher viral vector production yields. Cells grow on the mesh as a monolayer, and as we pack these, we reach a surface density of approximately 100,000 square centimeter per liter or 10 square meter per liter in the current design. What is key again is the uniform fluid distribution throughout the column as cells are uniformly exposed, including to transfection reagents. They get access to nutrients, eliminate waste, and are basically growing in a healthier environment, leading to better performance. Let's now take a closer look at the flow of the fluid through the bioreactor. We present it here in two ways, the theoretical modeling and the experimental results. On the left, you can see that the colors represent the velocity of the fluid, with red indicating high velocity and high shear, and green representing low velocity, low shear. Within the setup of our system, media enters the bioreactor vessel at high velocity, where it encounters a specifically designed distribution plate to uniformly disperse fluid flow. Flow through the bioreactor vessel is slow, low shear and uniform till it leaves first the mesh bed, then gains velocity again upon leaving the reactor. In the middle, you can see a graphical representation of the actual flow through the bioreactor, the experimental data. They show a sharp peak, which indicates a rapid and uniform movement of the fluid band from the top to the bottom of the bioreactor vessel. Both modeling and experiment validate the goals. Efficient nutrient and reagent delivery, waste removal throughout the bed, but also homogeneous cell attachment, growth, and transfection. To further demonstrate uniformity, we sample 4 discs from the indicate level of a 4 square meter bioreactor vessel, 3 for staining by crystal violet and 1 for cell counting. You can see a very uniform distribution of cell growth across each of the mesh layers from top to bottom. And upon harvest and cell counting, we show a similar indication of uniformity bottom to top of the vessel. Normalizing cell count to the top layer shows an average of 1.44 difference in cell count through the height of the vessel. In the table to the right, we show the scalability of the bioreactor vessel design by showing the cell yield per square centimeter for the reactor, varying in total surface area or number of mesh layers. When grown under similar conditions, the yields are very consistent. Let me now show you some data on the ability to harvest cells from the mesh. The 3 columns on the left here show the cell coverage before cell removal by enzymatic dissociation, and the 3 columns on the right show mesh stain after cell removal, indicating over 90% cell release and recovery. In addition, harvested cell population are typically over 95% viable. To close on data, I want to highlight 2 features: the higher yield of AAV per surface area, as well as the capability of the SN FBR to enable uniform cell transfection. On the left table, we have compared the AAV yield of our platform compared to the incumbent FBR. The results show a 4.5-fold reduction in number of vessels, a 3-fold higher yield per surface area, 33% less media consumption, and a 2.5-fold lower virus harvest volume. The right panel shows the transfection data. The left histogram shows the percentage of cells transfected by GFP plasmid in the entire FBR as determined after transfection cell harvest. And the right shows the transfection efficiency to the top, middle, and bottom of the vessel, indicating the uniformity of the in situ transfection process. Let me now summarize the technology and the product features and benefits in comparison with other competitive fixed bed reactor or suspension technologies. As demonstrated briefly here today, our Corning Ascent solution has been designed to maximize homogeneous cell growth, which leads to increase in viral vector yields, and importantly, allows cell recovery in adherent format. And these 2 top attributes, distinguish our FBR system from competition. We believe the linear scalability of the system and the broad range of potential applications is a further advantage of our technology. In summary, I have presented to you our novel Corning Ascend fixed bed reactor solution for scale-up that will be launched shortly. On the process development unit, we and our pharma partners have demonstrated improved viral vector production yields due to our innovative fixed bed reactor design. We have shown viable cell harvest and transfection in adherent format. Furthermore, the unit enables reduced media usage and lower volume of harvest. And finally, we're currently working on expanding the scope of this technology beyond viral vector production, as a cell harvest feature enables areas such as stem cell work or clean meat to be potentially addressed. If you want to learn more about our technology, please contact me directly at the email address shown on the slide. Again, thank you for your attention, and I hope you enjoy the rest of the conference.